Let's start with the anatomy of the flowering plants. Chapter 6, Anatomy of the flowering plants. Here we are going to see about the tissue system, anatomy of dicotyledonous and monocotyledonous plants. You can very easily see the structural similarities and variations in the external morphology of large living organisms, both plants and animals. Similarly, if you were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to internal structure and functional organization of higher plants. Study of internal structures of plants is called as anatomy. Plants have cells as the basic unit. Cells are organized into tissues, and in turn, the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. Basically, about why we should study anatomy. In anatomy, we study the internal structure. Normally, when we see morphology, we can easily identify structural similarities, variations, etc. Anatomy also shows such variations, such as within angiosperms, monocots and dicots are seen to be anatomically different. Now, the first is the tissue system. We were discussing types of tissues based on types of cell present. Let us now consider how tissues vary depending on their location in the plant body. The structure and function would also be dependent on the location. On the basis of their structure and location, there are three types of tissue system. There are epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. Okay, basically in plants there are three types of tissue system. These are based on the function they perform, their structure, and their location. The first one is epidermal tissue system. Second one is the ground or fundamental tissue system. Third one is our vascular system. Now let's go to the epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. Okay, what they are saying is the epidermal tissue system, this tissue system is everything that covers the plant along with trichomes, that are the hairs, and epidermal appendages, so that are trichomes, and basically the whole plant coverings, that is epidermis. We can call it the skin of the plant. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. The epidermis is the outermost layer of the plant body, the primary plant body. This is a very elongated and compactly arranged epidermal cells. These form continuous layer. This epidermis is usually single layered. Epidermal cells are parenchymatous with small amount of cytoplasm lining the cell wall and a large vacuole. These epidermal cells are basically parenchyma cells, a type of permanent tissue. We know that tissues are divided into meristematic, non-meristematic, and under non-meristematic there are simple and complex. Under simple non-meristematic tissues, we have parenchyma, collenchyma, and sclerenchyma, out of which parenchyma and collenchyma are living cells and sclerenchyma is dead cell. Here, the epidermis is made up of parenchymatous cells. They have small amount of cytoplasm and large vacuole. The outside of the epidermis is often covered with a waxy thick layer called as the cuticle, which prevents the loss of water. Cuticle is absent in roots. Okay. The epidermis outer layer is covered by a thick waxy thick layer. This layer is called as the cuticle and it prevents water loss through transpiration. It's not present in the root obviously, because if it was present in the root, how would the water go into the roots? Stomata are structures present in the epidermis of the leaves. Stomata regulate the process of transpiration and gaseous exchange. Okay, stomata are structures present in the epidermis of the leaves. We know about stomata, the pore-like structures present in the leaf. And they regulate the process of transpiration, loss of water, and gaseous exchange. Each stoma is composed of two bean-shaped cells known as the guard cells, which enclose the stomatal pore. The stomata consists of two guard cells, bean-like cells, which enclose the stomatal pore. In grasses, the guard cells are dumbbell-shaped monocots. The guard cells are dumbbell-shaped. In case of dicots, they are bean-shaped. The outer walls of guard cells away from the stomatal pore are thin and the inner walls towards the stomatal pore are highly thickened. The outer walls are thin, but the inner walls, that is towards the stomatal pore, which they need to cover, at that side they are very thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. These guard cells possess the chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cell become specialized in their shape and size and are known as subsidiary cells. Okay, surrounding the stomata and the guard cells, there exist few epidermal cells. They are epidermis only, but they become specialized in their shape and size, and they are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells all together are called the stomatal apparatus. There are three main things in the stomatal apparatus: the stomatal pore, the guard cells, and the subsidiary cells. As for what subsidiary cells are, there are cells surrounding the guard cells. The epidermal cells surrounding the guard cells get specialized in their shape and function, and they become the subsidiary cells. All together form the stomatal apparatus. Epidermal cells, this is the stomatal pore. This is with bean-shaped guard cells, mostly in dicots, and dumbbell-shaped in monocots. Diagrammatic representation of stomata with bean-shaped guard cells, stomata with dumbbell-shaped guard cell. This is the epidermal cells, and these are the epidermal cells which turn into subsidiary cells. Chloroplast in the guard cell here, and this is the stomatal pore finally. This is the stomatal pore here as well, dumbbell-shaped cells. Subsidiary cells are here, epidermal cells are here, and the guard cells are here, the dumbbell-shaped guard cells. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called as trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. The epidermis possesses hair-like structures. There are two hair-like structures. One we see at the root epidermis, where we see there are root hairs. They are mostly unicellular root hairs that help in absorption of water and minerals. And in the above layers, in the shoot system, we see Trichomes, they are also hairs, but they are multicellular. They can be branched, stiff, or unbranched, or soft. Their main role is to prevent or reduce the loss of water due to transpiration. Now let's go to the second kind of system, the ground tissue system or the fundamental tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. Except epidermis and vascular bundles, whatever there is in the plant, all comes under the ground tissue system. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary rays in the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called as the mesophyll. Okay, first point here, all tissues except epidermis and vascular bundles are called as ground tissue, and this ground tissue consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. And parenchymatous cells, these where we find these parenchymatous ground tissue cells, we usually find them in the cortex region, pericycle region, pith region, medullary rays, and in some primary stems and roots. In leaves, the ground tissue, in case of leaves, this ground tissue is made up of thin-walled chloroplast containing cells. These are called as the mesophyll cells. Now next is the vascular tissue system. Let's see the diagrams first. Various types of vascular bundles: radial, conjoint closed, and conjoint open. This is the a radial type of arrangement. Basically, this is xylem here, this is xylem, and these are phloem. They are arranged in this manner, in this fashion we can say. This is called as the radial arrangement of the vascular bundles. And the next one is conjoint arrangement of the vascular bundles, where they are joined together, xylem and phloem. This is conjoint closed and conjoint open. As for why one is open, why one is called closed, is because of the presence of cambium. Cambium is a tissue which will help in secondary growth, the growth of the width of the plant, and if it is there, there will be a growth in the width of the plant. If it's not there, in case of monocots, there will be no width growth of the plant. So here it is called as closed because there is no cambium, and here it is called as open because there is a presence of cambium. The vascular system consists of complex tissues, the phloem and the xylem. Under the non-meristematic tissues, there are simple and complex. Under the complex tissues, we have xylem and phloem. These xylem and phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. Okay, this is in case of dicotyledonous plants. In monocots, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed. In case of open systems, we can, as the distance separates, new xylem and phloem tissues are formed. But in case of monocots, there is no such thing. Absence of cambium. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. Okay, what they are saying is the conjoint closed and open we saw. As for the radial, when xylem and phloem, these two tissues, are arranged in an alternate manner, like here if we see first xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem. There is an alternate manner. Along the different radii, the arrangement is called as radial. This is seen in roots. In conjoined type of vascular bundles, the xylem and phloem are jointly situated along the same radius of the vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. Okay, if first thing there are different radii, alternate arrangement is radial arrangement seen in roots, or conjoined, same radii, same levels joined together is called as conjoint, and it is seen in stems and leaves. It can be open or closed. Now let's go to the next topic, anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stems, and leaves, it is convenient to study the transverse section of the mature zones of these organs, that is, stems, leaves, and roots. Dicotyledonous roots, that is first diagram. Look at figure 6.3. Let's see the figure first. 6.3a, yes. Okay, this is the monocot root and this is the dicot root. If we see the figure here, this is the first layer here, starting with epidermis here, epidermis, root hairs here help in absorption of water and minerals. This whole thing is the cortex region, and next is the endodermis approximately here, endodermis. Then comes the pericycle, this one here, not sure if it's visible here, this one is the pericycle, this one is the epidermis above the pericycle, and these ones are the protoxylem, protoxylem here, pith is here, and phloem it is here. Metaxylem is here. Basically that's with the structure of the dicot root. This is the radial arrangement we saw. They are arranged radially here in case of roots. Let's see the monocot root as well. Nothing much different. Roots are there. Epidermis is there, cortex, endodermis, pericycle, phloem, protoxylem, pith, and metaxylem. Almost everything same, just more number of bundles, more different arrangement of the different order of succession of the uh, xylem and phloem. Basically, that one we will see right now. It shows the transverse section of this sunflower root. The internal tissue organization is as follows. The outer layer is epiblemma. Many of the cells of epiblemma protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called as endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. Okay, basically, epiblema is the outermost layer. Instead of calling it epidermis, they call it epiblema here. And there are root hairs, unicellular. Then after that, we have cortex. Cortex is parenchymatous cells, which are having more number of intercellular spaces. Then the most innermost layer of the cortex is called the endodermis. Endodermis has is made up of barrel-shaped cells. There are no intercellular spaces in case of endodermis. The tangential as well as radial walls of the endodermal cells have a disposition of water-impermeable waxy material suberin in the form of Casparian strips. Basically, these endodermal cells, the walls of the endodermal cells, which are cylindrical in shape, have a deposition of waxy material that is called as suberin, and this is in the form of Casparian strips. If there is a question which cells contain the waxy layer suberin deposition and form Casparian strips, it is the epidermal cells, endodermal cells. The walls of the endodermal cells have a deposition of a water impermeable waxy material called as suberin. It is in the form of Casparian strips, and next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Pericycle is made up of parenchyma. Epidermis is made up of parenchyma as well. Cortex parenchyma as well. Endodermal cells, I'm not too sure, it's not mentioned here. Then pericycle is parenchyma as well. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pericycle, which is the lateral growth initiation, we saw cambium helps in lateral growth initiation. This initiation of the secondary growth starts at the pericycle cells. And in case of dicotyledonous root, the pith is small and inconspicuous. The parenchymatous cells, which lie between the xylem and phloem, are called as conjunctive tissue. Okay, the parenchyma cells that is lying, these cells lying between xylem and phloem, those are called as conjunctive tissue. There are usually two to four xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side and the endodermis, such as pericycle, vascular bundles, and pith, constitute the stellar region. Okay. Basically, the tissue between the xylem and phloem is called as conjunctive tissue. Next is there are, in case of dicotyledonous root, there are 2 to 4 xylem and phloem patches, and all tissues on the inner side of the endodermis. If endodermis is a wall, everything inside that wall, close to that wall, is called as stellar region. That constitutes the vascular region, pericycle, pith, all constitute the stellar region inside the endodermis. Now let's go to the monocot root. The anatomy of monocot root is similar to the dicot root. In many respects, it has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith, as compared to the dicot root, which had fewer xylem bundles. There are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. Okay. Everything is mostly similar to the dicot root, but what changes here is the amount of xylem bundles, xylem and phloem bundles. In case of the dicot, we saw it was 2 to 4, but here we see 6. This is a polyarch situation. And here, one thing more is their pith was inconspicuous and small, but here pith is large and well developed, and there was lateral growth. Here in monocots, there was no secondary or lateral growth. Now let's go to the next main topic, that is dicotyledonous stem. Let's see the diagram first. The first one is dicot, second one is monocot. This is the epidermis here. Epidermis trichomes or epidermal hair. Next constitutes collenchyma for mechanical support. Then comes parenchyma. Then is the innermost layer, that is endodermis. Then pericycle, phloem, cambium, and metaxylem, protoxylem, and pith. Pretty similar here. Both are same thing, just transverse section and vertical section, or a magnified picture here. Here the first thing is epidermis, second is collenchyma, third is parenchyma, fourth is endodermis, fifth is pericycle, sixth is phloem, seventh is cambium. First one epidermis, second one collenchyma, next is parenchyma, endodermis 4, pericycle 5, phloem 6, cambium 7 for open vascular bundles, that is in dicots, and 8 metaxylem, 9 protoxylem, and 10 apith. Total of 10 parts. There is a hypodermis here, that is below the epidermis. Parenchyma, endodermis, pericycle, vascular bundle, and medullary race here. Now in case of monocot, if we see, there is the epidermis here and here. Then below the epidermis is the hypodermis. Next is the ground tissue. Everywhere is the ground tissue, and randomly there are face-like structures here, like this is circle face-like structures. These are the vascular bundles consisting of a xylem and a phloem, and remaining everything is ground tissue, this green color. Everything other than the red color face-like structures are vascular bundles. There are many vascular bundles in monocots here. That's basically the diagram. Not very complex, it's similar to the roots, just different arrangement. Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in multiple layers between the epidermis and pericycle constitute the cortex, similar to before. It consists of 3 subzones. The outer hypodermis constitutes a few layers of collenchymatous cells just below the epidermis, which provides mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of cortex is called the endodermis, same as before. The cells of the endodermis are rich in starch grains, and the layer is also referred to as the starch sheath. Okay. In case of roots, the endodermis cells were cylindrical and had deposition of suberin and made the Casparian strips. Here, in case of dicot stem, everything is similar with epidermis cells constituting trichomes, and below the epidermis there is hypodermis, which is the collenchymatous cells, and multiple layers between epidermis and the pericycle. This constitutes the cortex, and this cortex constitutes of 3 zones, that is hypodermis, the collenchymatous cells, and parenchymatous cells below the hypodermis, cortical layers that we call, of rounded thin-walled parenchymatous cells with conspicuous intercellular spaces. Then a part of cortex, the third part of cortex, is endodermis, which is having a large amount of starch grains, and this is called the starch sheath. Pericycle is present on the inner side of endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Okay, they're saying that pericycle is actually present on the inner side of the endodermis, like similar to roots. It is present in the inner side of the endodermis. Pericycle is there, and it is present above the phloem tissue. It is present in the form of semilunar patches, that is of sclerenchyma tissue. Pericycle is made up of sclerenchyma tissue. It is in the form of semilunar patches. Remembered, point to be remembered. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute medullary rays. Okay, pericycle is present in the inner part of the endodermis, and these are from the sclerenchyma cells. Next to that, in between the vascular bundles, there are few layers of radially placed parenchymatous cells. In between the vascular bundles, like here, if we observe, these are the vascular bundles. These two, otherwise, let's assume these two are vascular bundles. Then this space, this yellow color space between them, which constitutes of parenchymatous cell, called medullary rays. Here, if we see medullary rays are made up of parenchyma cells. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute the medullary layers. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of a dicot stem. It is a point to be remembered. A ring arrangement. If we see above that vertical section, this is the ring arrangement of the vascular bundles. It is a characteristic feature of the dicot stem. If we see in the monocot stem, we do not see such arrangement. They are arranged as circles or faces. Each vascular bundle is conjoined open with endark protoxylem. A large number of round parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. Okay, the vascular bundles are conjoined, they are open, there is cambium present between them, they are endark protoxylem. Endark protoxylem meaning, if we see the diagram here, the protoxylem are showed here. Endark meaning, let's just be sure about it. Endark protoxylem It's basically arrangement which come first and which come last. Okay. If protoxylem is, where is this diagram? Yes. Protoxylem is here. This is the old xylem that is first formed, and metaxylem is the latest xylem. So end arc meaning the newer one is inside, whereas the outer one is towards the outside. The newer one is outside. The protoxylem is located toward the center. That is the old one, and metaxylem lies towards the periphery. And the parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. Now monocot stem. Monocot stem has sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than central located ones. A point to be remembered statement here. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water containing cavities are present within the vascular bundles. Okay, let's see that again. The monocot stem has a sclerenchymatous hypodermis. In case of the dicot stem, we saw it was collenchyma, but here it is sclerenchymatous. Both are for mechanical support, but here sclerenchyma meaning dead cells, whereas the dicot had living cells. Here the vascular bundles are very large, very large in number, and they are scattered. Each of them, each of the vascular bundles is surrounded by a sclerenchymatous bundle sheath. The sclerenchymatous bundle sheath covers the each of the vascular bundles. Like here, these faces are covered by sclerenchymatous sheaths. And there is a large and conspicuous parenchymatous ground tissue. Whole thing other than the face-like structures is ground tissue only. So it is large parenchymatous ground tissue. These vascular bundles are conjoined and closed. They are one thing different, it is closed. They don't have cambium. And if we go from center to outside, the center vascular bundles we see at the center are generally larger than the outside. These vascular bundles here are larger compared to these ones here, which we can see actually here. That is what they're saying. The phloem parenchyma is absent, and water containing cavities are present within the vascular bundles. Phloem parenchyma is a kind of tissue involved in the complex tissue of phloem, and they do not give much details about it, so I will not talk about it here either. Now this is the next one is the dorsiventral leaf, basically a dicot leaf. Why it's called a dorsiventral leaf? It's because the two sides of a dicot leaf are different, and the dorsal side is different from the ventral side. That is why it's called a dorsiventral leaf. In case of a monocot leaf, we call it as a isobilateral leaf because both sides of a monocot leaf are same. That's why the word iso came there. Let's first see the dorsiventral leaf first. This is the dorsiventral leaf structure, a schematic representation here. There are two surfaces for the dorsiventral leaf, the adaxial surface and the abaxial surface. The adaxial is the outer surface which is exposed to the sunlight. Abaxial is the below surface. The outermost layer is the adaxial epidermis. There is a bundle sheath here that is surrounding the vascular bundles that is made of sclerin, or maybe not. It is dicot here, so I'm not sure. Xylem phloem, bundle sheath cells and bundle sheath. Stomatal pore here. And the main thing, the star of the show, is the palisade mesophyll and the spongy mesophyll here. These both are the main additions here compared to the root and shoot, that is the mesophyll tissue. These are the cells which capture the sunlight and do photosynthesis. The palisade mesophyll are towards the sun, they are more concentrated, long, elongated, and they have more amount of chlorophyll to do perfect photosynthesis. They are the most efficient photosynthesizing agents in the leaf. And below that, the mesophyll tissue is the leaf tissue which actually undergoes photosynthesis. And out of that, there are two types, spongy and palisade, where palisade is the one long elongated, more number of chlorophyll, and they are the more efficient at photosynthesis. Spongy mesophills are below the palisade mesophyll. They are arranged in a way that there are more number of intercellular spaces, and they are irregular in shape. As for palisade mesophyll, they are long elongated, very minute intercellular spaces. They avoid intercellular spaces. And if we see above, there won't be much concentration of stomata in the adaxial surface. There will be more stomata in the abaxial surface only. There will be less amount of stomata in the adaxial surface, that is exposed to the sun. Only function is capture of sunlight. Below, where it doesn't have a function, it will have of capturing sunlight, it will have stomatal pores here below, more number of stomatal pores. That is why the upper side is comparatively darker because of the palisade and spongy mesophyll. If we see a dicot leaf, the two sides, the adaxial side will be comparatively a darker green compared to the below side, that is the abaxial side. That is because of the concentration of the palisade mesophyll. Now let's see the isobilateral leaf as well. This is the isobilateral leaf. Here both sides are equal. There is no differentiation between mesophyll tissue. There is no palisade. There is no spongy mesophyll. There is adaxial and abaxial surfaces. Both are same. There is the xylem mesophyll tissue. The mesophyll tissue is also same. There are stomata on the both sides. And there is a normal everything that you see. Now let's see the paragraph they have given. The vertical section of the dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface, adaxial epidermis, and lower surface, abaxial epidermis, of the leaf, has a conspicuous cuticle to prevent transcription or to avoid water loss. The abaxial epidermis generally bears more stomata than the adaxial epidermis. That's what I said. There are more number of stomata in the below surface compared to the above surface. The latter meaning the adaxial surface may even lack stomata. The upper surface may completely even lack stomata as well. The tissue between the upper and the lower epidermis is called the mesophyll tissue. Mesophyll, which possesses chloroplasts and carry out photosynthesis, is made up of parenchyma. This is a parenchyma, that's what we saw before. It has two types of cells, the palisade parenchyma and spongy parenchyma or spongy mesophyll. The adaxially placed, the upper placed palisade parenchyma is made up of elongated cells which are Vertically parallel to each other, the oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system indicates vascular bundles which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. Okay, let's see the before statement. There are numerous very large spaces and air cavities between the spongy mesophyll cells. Okay, now vascular system includes vascular bundles. Okay, which can be seen in the veins and the vein midrib. In the veins, the function of veins and veinlets is to transport minerals and water and to provide rigidity to the lamina. The first function, to transport minerals and water, that is the reason it contains vascular bundles. The size of the vascular bundles are dependent on the size of the veins. More the number of the veins, more the number of the veinlets, larger the radius of the veins and veinlets, the larger the size of the vascular bundles. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick walled cells, the bundle sheath cells. Look at figure 6.1 and find the position of xylem in the vascular bundle. This is a xylem surrounded by bundle sheath cells. Now the isobilateral leaf. The anatomy of the isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. Only we are going to see about the differences. Isobilateral leaf, the stomata are present on both the surfaces of the epidermis. The mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermis cells along the veins modify themselves into large, empty, colorless cells. Okay, in grasses, if we see, okay, first difference is there are no differentiation palisade and spongy. There are only mesophyll cells. That's all. Only mesophyll, no differentiation. And next information, in grasses, if we see, certain adaxial epidermal cells along the veins, there will be certain adaxial epidermal cells, above surface epidermal cells. These cells along the veins, they get modified. What they get modified into? They get modified into large, empty, colorless cells. These colorless, empty cells are called as bulliform cells. When the bulliform cells in the leaves have absorbed water, we know that they're empty. That's what they said. So of course they can absorb water if they're empty. Because of they are empty, they will absorb water. When they absorb water and are turgid, the leaf surface is exposed. They become rigidity and expose the leaf surface. When they are flaccid due to water stress, meaning less water, they make the leaves curl inwards to minimize the water loss. When they have a large amount of water, there will be large amount of water which will provide turgidity to the leaf surface, and the lamina of the leaf is exposed. Because of that, the sunlight will touch the leaf and it will undergo photosynthesis, because there is no lack of resources, and it will start photosynthesis. But if there is lack of water, then the bulliform cells will not have water to get filled. At that time, due to lack of water, the leaf will curl over. And the advantage of this is that when there is no water, the leaf won't be exposed to the sunlight. Due to this, there won't be any loss due to transpiration. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles. Okay, like we can identify the sizes of the vascular bundles based on the venation size. In case of parallel venation, monocot leaves, like the veins will be same size as the vascular bundles. except in the main veins, as seen in vertical sections of the leaves. Summary here. Anatomically, a plant is made up of different kind of tissues. The plant tissues are broadly classified into meristematic, that is apical, lateral and intercalary, and permanent, simple and complex. Assimilation of food and its storage, let's just mark it, there's extra information that is not given in the lesson, which we should know basically. The plant tissues are broadly classified into meristematic, apical, lateral and intercalary meristems, and permanent, that is simple and complex meristem. Assimilation, usage of food and its storage, transportation of water, minerals and photosynthesis, and mechanical support are the main function of tissues. The functions of tissues is assimilation of food and its storage, transportation of water and minerals and photosynthesis, food also transport, and mechanical support are the main functions of tissues. There are three types of tissues: epidermal, ground and vascular. The epidermal tissue systems are made up of epidermal cells, stomata and the epidermal appendages. The ground tissue system forms the main bulk of the plant. Ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle and pith. The vascular tissue system is formed by xylem and phloem on the basis of presence of cambium, location of xylem and phloem. The vascular bundles are of different types. The vascular bundles from the connecting tissue and translocate water, minerals and food materials. That is, xylem transport water and minerals, phloem transports food. Monocotyledonous and dicotyledonous plants show marked variation in their internal structures. The different type, number, and location of vascular bundles. The secondary growth occurs in most of the dicotyledonous roots and stems. First revision of this chapter via NCERT completed.